Good evening, everybody. How you're doing today? Today we are discussing a quick review. We're gonna have a quick review of the content of the first practical exam. Good evening, everybody. How you're doing? Today we're gonna have a quick overview of the material covered by the first lab exam. First important diagram that we have on here is going to be the animal cells, the general structure of the animal cells. This is a picture of a model that you have on campus. So what we're looking at here, number one, is this set of tubules that is going to be without any kind of dots, those whitish dots on the outside. We don't have any of them on here. And I am directly connected to either the nuclear membrane or to the rough endoplasmic reticulum. This is my Golgi body or the Golgi apparatus. Golgi apparatus is going to be responsible for the concentration, the trimming of the proteins, and the packaging of the proteins. Looking at number two on here, number two is pointing to the rough endoplasmic reticulum. And you see on here, this is another set of tubules, but on the outer surface of those tubules, we have those white dots on the outside. Those white dots are the ribosomes. The main function of the ribosomes, along with the rough endoplasmic reticulum, is going to be for the production of the protein or protein synthesis. This is my main function on here. Looking at number three on the side, just close to the center of the cell. Number three, it's a set of tubules. As you see on here, this set of tubules, those are the centrioles, and I have two sets of tubules. Two centrioles together will form my centrosome. So if I'm looking here, close, one set is the centriole, the two sets together, this is my centrosome. I have another cluster. Before we move on to the smooth endoplasmic reticulum, the function here of the centrioles along with the centrosome, it's the formation of the mitotic spindle fibers. The mitotic spindle fibers, as we're going to discuss in a minute, it's going to be responsible for the separation of the chromosomes during the cell division. So it's really important to complete the cell division, allowing the cell to split its genetic material into two different sets. Moving on to another organelle in the cell, it's going to be the smooth endoplasmic reticulum. And again, this is going to be a set of tubules, membranous tubules. And I don't have any kind of ribosomes on the outside. This is marked by number 5. The function of the smooth endoplasmic reticulum is going to be determined according to what kind of cell is it located in. So for example, the smooth endoplasmic reticulum in the liver cells is going to be responsible for the metabolism of the fat, for the detoxification of the toxin and medications. In, for example, the muscle cells, it's going to be responsible for the storage and release of the calcium, which is needed for the muscle contraction. Moving on to another organelle, another membranous organelle on here is going to be this vesicle. This actually, you can't just recognize what's the content of the vesicle, but we're gonna keep in mind that this specific vesicle is the peroxisome. And the peroxisomes are vesicles that contain oxidative enzymes, and they will be responsible to detoxify some of the free radicals that will be resulting from the metabolic cellular activities. So this vesicle, it contains oxidative enzymes, and again, we call this is my peroxisome. I have a double membrane organelle, many of them. So we've got 1, 2, 3, 4 of them on here. This double membrane organelle, it's the mitochondria. And the mitochondria is a powerhouse of the cell. It's going to be responsible for the formation or the production of ATP, which is the cellular energy. This is a form of energy that your cell can deal with. Moving on to the outer barrier that would be separating the genetic material inside the nucleus from the rest of the organelles in the cytoplasm on the outside. This is my nuclear membrane. At the center of the nucleus, I have the nucleolus, and the nucleolus is a part of the cell that is going to be responsible for the production of a special type of RNA. We call this is the ribosomal RNA or rRNA. The ribosomal RNA, it enters in the formation of the ribosome. So in order for me to produce those ribosomes, I need to form a ribosomal RNA, and this can only be produced within the nucleolus of the nucleus. If you see on here, number 10 is pointing to another vesicle that is fusing with the cell membrane. So we call those vesicles are the secretory vesicles. So they contain, for example, a protein that acts as a hormone or that acts as an enzyme, and now the cell is ready to release it to the outside. As you've learned back in chapter 3, that this type of transport in which you are releasing the vesicular content to the outside, it's called exocytosis. So the secretory vesicles are going to allow exocytosis to take place. Another type of vesicles that we can see inside the cytoplasm of the cell is marked as number 11. It's the lysosomes. Lysosomes are going to be responsible to break down things. Remember, lysis means break down, and om means body. So this is the body that would be breaking down. This vesicle contains digestive enzymes, and the digestive enzymes are going to be required for your immune cells to break down bacterial cells. It's going to be breaking down non-functional organelles. It's going to be actually, in some cases, breaking down itself. So in some cases, a cell might decide to destroy itself because it's non-functional. It's going to be performing what we call autolysis. Auto means self, lysis, to lyse is to break down. So autolysis means self-breakdown. So in some cases, these cells might be using those digestive enzymes stored inside the lysosomes to break down themselves. Number 12 here is pointing to the part of the cytoplasm that is not containing any organelles. This is the part that we call the cytosol. So cytosol is the matrix where the organelles are going to be located. If I'm looking here at number 13 is going to be marking the outer boundary of the cell. And if you remember, what separates the internal environment of the cell from the external environment of the cell, it's going to be the cytoplasmic membrane. Looking at another diagram showing the same exact organelles. Those two diagrams are highly important for your upcoming lab exam. So we're looking here, mitochondria, membrane organelle. Again, it's going to be responsible for the ATP production or the energy production. The plasma membrane or the cytoplasmic membrane, it forms the outer boundary of the cell. I have the centrioles that are forming together the centrosome. I have vesicles that contain digestive enzymes. Those are my lysosomes. I have those sets of tubules that won't have any kind of ribosome to the outside of their outer surface. Those are my smooth those are forming my smooth endoplasmic reticulum. And if you remember, smooth endoplasmic reticulum again was responsible for metabolism of the fat in the liver cells, in the intestinal cells. It's going to be responsible for detoxification of the toxins, medications in the liver and the kidney. It's going to be responsible for storage and release of the calcium for muscle contraction. And this is going to be taking place in the skeletal, cardiac, or smooth muscle cells. So you see here, smooth endoplasmic reticulum, again, its function is going to be determined according to what organ cell is it located in. Secretory vesicle, again, it's fusing with the plasma membrane to allow the release of the vesicular content to the outside of the cell by exocytosis. I have here other types of vesicles that this time will contain oxidative enzymes. The oxidative enzymes here are meant to reduce the oxidative enzymes are going to be responsible for the detoxification of the free radicals to transform them into more stable molecules, something like the transformation of the free radical here, hydrogen peroxide, into something like the water. Something less toxic, something that is chemically much more stable. What if the cell contains fat, for example, like the fat cell? Where is this fat kept? Inside the cell. This fat is simply kept inside larger vesicles. We call those large vesicles that contain those inclusions are the vacuoles. The part, again, of the cells that contains the matrix, the fluid, and the inclusions, as well as the organelles, all this together is what we call the cytoplasm. It's gonna be all the space between the cytoplasmic membrane and the nuclear membrane of the nucleus. So anything in between, it's gonna be part of the cytoplasm. Looking here at this set of tubules, I don't have any ribosomes on top of me. This is my Golgi apparatus. And again, the function of the Golgi apparatus would be to contract, to trim and package the proteins before reaching their destination. Looking on the inside of the nuclear membrane, I will see the DNA of the cell is going to be kept in the form of chromatin. At the center of the cell, I have a dark structure on here. This is my nucleolus. And I will see here all the structure all together, nuclear membrane, chromatin, and nucleolus. All this will be forming my nucleus. I will see connected to the nuclear membrane of the cell, I will see sets of tubules, and on the outer surface on them, I have those red dots. Those are my ribosomes, and this set of tubules that has the ribosomes on its outer surface, this is my rough endoplasmic reticulum. Remember, ribosomes can exist either attached to the tubules of the rough endoplasmic reticulum. We call those are membrane-bound ribosomes, and those are going to be responsible for the production and the formation of the proteins that will either be released from the cell or that will be integrated within the plasma membrane. Looking at two parts of the cytoskeleton of the cell, I have the intermediate filaments on here, and I have much thicker filaments or tubules that will also enter in the formation of the skeleton of the cell. We call it the cytoskeleton. It's going to be the microtubules. So we've got here two examples of the filaments that build up my cytoskeleton. A quick review of the cell cycle. If you remember, the cell cycle is simply the life cycle of a cell, and it's going to be divided into two main phases. The first phase on here, it's going to be the interphase, and the second phase, it's going to be the mitotic phase. Interphase is subdivided into three phases: G1 or growth 1, S or synthesis, and G2 growth 2. So during interphase, the cell will be growing, will be increasing the amount of cytoplasm, will be increasing the number of organelles, will duplicate its genetic material, and the duplication of the DNA is going to be taking place during the S phase of the interphase. So interphase is simply the time of when the cell is preparing for the cellular division. During the mitotic phase, the cell is going to be undergoing the cell division. Mitotic phase is going to be divided into two main parts. First part is going to be the mitosis, and the mitosis is the nuclear division. This is when the nucleus of the cell is divided into two nuclei. And the second part of the mitotic phase, it's going to be the cytokinesis or the cell division. So very important to differentiate between mitosis and cytokinesis. Mitosis means nuclear division. Cytokinesis is the cell division. Both together, it's the mitotic phase of the cell cycle. So the cell during interphase, this is how it's going to look like. The DNA is going to be in a chromatin form. You're going to have a nuclear membrane for the nucleus. You're going to have the nucleolus, and you're going to have two sets of microtubules. Those are the centrioles forming the centrosome. When the cell starts its mitotic division, it passes through the first phase, which is my prophase. What happens during prophase? The chromatin here, the DNA, and the genetic material is going to be condensed to form chromosomes. Each chromosome going to have two sister chromatids attached to one another by the centromere. Another thing that will be taking place is the degeneration of the nuclear membrane. The nuclear membrane disappears as well as the nucleolus. So I don't have any more nuclear membrane. I don't have any more nucleolus. And again, the chromatin here has been condensed into chromosomes, as you see. The centrioles of the centrosome gonna be moving to the two opposing poles of the cell, as you see on here. Those are the centrioles of the centrosome, and they will be forming the mitotic spindle fibers that will be attaching the chromosomes at the centromere. All this is going to be taking place during prophase. The mitotic spindle fibers will start to contract, pulling the chromosomes towards the two opposing poles of the cell. This will cause the arrangement of the chromosomes and their centromeres at the equator of the cell, forming what we call the metaphase plate. And this is the phase when I will be performing, this is a phase that I call here my metaphase. As the mitotic spindle fibers keep contracting, pulling the two sister chromatids away from one another, this is gonna cause the separation of the two sister chromatids. And as I keep pulling on them, you see on here, if you are pulling on a towel, for example, from its center, what's gonna happen? It's gonna give you a V-shaped appearance. This is exactly the same things that will be taking place for those chromosomes that you have separated, moving them towards the two opposing poles of the cell. By this, I will be forming two sets of chromosomes on each side of the cell. During the last phase of the mitosis, which is my telophase, those chromosomes that I've moved to this pole of the cell will be returning to the chromatin structure. The nuclear membrane will reappear, as well as the nucleolus, and this will be taking place in the two sides of the cell, forming two separate nuclei inside the cell. This is my mitosis. How would I be separating those two nuclei, forming two cells? This is going to be my cytokinesis. And as you see on here, I form a constriction ring that will keep widening, and the two sides will fuse together, separating the two sides of the cell, forming two daughter cells, which will resemble the original mother cell. So looking at the diagrams on here, those are pictures taken from the models that you have on campus. So what is this showing? I see here, this is my nuclear membrane. This is my cell membrane, cytoplasm. Then on the inside, I have the DNA inside the nucleus, and it's going to be in the form of chromatin. This is my interface. Starting the mitosis, you see the nuclear membrane is going to be broken down, nucleolus will disappear, the centrioles are going to be migrating to the two opposing poles of the cell, forming their mitotic spindle fibers. The mitotic spindle fibers will be attaching themselves to the centromere of the chromosomes and will start pulling on them, and this will allow the chromosomes to be arranged at the equator of the cell. And this phase is what we call the metaphase. As I keep pulling those two sister chromatids away from one another, again, this is gonna cause the rupture of the centromere, attaching them together. And I will be able to form two sets of chromosomes on both sides of the cell. the phase in which I have the separation of the two sister chromatids from one another, forming two sets of chromosomes. It's called my anaphase. Each set of chromosomes will have its own nuclear membrane, will return back to its chromatin shape, and the constriction ring here that separates the two sides of the cell will keep forming, resulting in the formation of the two daughter cells. This last phase of the mitosis is my telophase, and the separation of the two sides of the cell to produce two daughter cells that will be resembling the mother cells that we've started with. It's called cytokinesis. So very, very important again is to identify the different phases of the mitosis as well as the cytokinesis. Less important for us, don't worry about those diagrams, have a look at them, but those are diagrams that I won't be getting for you on the test. So very, very important to have a look at them, to have a better imagination of what's going on, but it's very unlikely that I will get you any of this on your exam. This table is showing you the different stages of the mitosis and cytokinesis, so please have a close look at the steps of the mitosis and cytokinesis for you to be able to identify them whenever you get one of those diagrams, for example. Those are much better to understand compared to those ones, so please stick to those ones on here. Those might appear on the exam. I will be asking you to identify the cell cycle phase. But as we have been discussing the different types of tissues, if you remember the type of tissue responsible for the formation of the coverings and linings of the organs, this is what we've called the epithelium. And we did have two factors according to which we classify the epithelium. If you remember, first, according to the number of cell layers, we classify the epithelium into simple or stratified. Simple epithelium means it's formed by one cell layer. If it's stratified, means it's formed by more than one cell layer. Another factor that we're using to identify the epithelium that forms the coverings and linings of the organs, it's going to be according to the shape of the apical cells. If I have flat cells, those cells are going to be squamous, and the tissue is going to be classified as squamous epithelium. If the apical cells are cube-shaped cells, the epithelium is going to be classified as a cuboidal epithelium. If the apical cells are tall cells, like what you see on here, it's going to be, the tissue is going to be classified as a columnar epithelium. So if I have one row of flat cells like what I see on here, it's going to be a simple squamous epithelium. If I have one row of cuboidal cells, it's going to be simple cuboidal. One row of columnar cells is going to be simple columnar. Many cell layers in the apical cells will be squamous or flat in shape. This is going to be a stratified squamous epithelium. If I have more than one cell layer and the apical cells are cube-shaped, this is going to be classified as a stratified cuboidal. We don't have actually stratified cuboidal as we're going to see in a second. So looking first at the simple epithelium, I have space, and this space is lined by layers of cells. Only one cell layer is lining this space, which is the air sacs in here in this example. And if I get closer, you will see that the cells are actually squamous. So I have flat cells on here that are forming this type of epithelium. So if I have one row of flat cells, it's a simple squamous epithelium. If you remember, simple squamous epithelium is going to be responsible to allow diffusion, is going to be responsible to form the lubricating substances in the serosa. Is going to be found in the air sacs of the lungs. Is going to be found lining the chambers of the heart, blood vessels, and the ventral body cavity. Look at on here, I have a space, and this space is lined with cells. You see one row of cells, and the cells here are cube-shaped cells. So what we're looking at in here, it's a simple cuboidal epithelium. Here is another example on here. You have space. The space is lined with cells, and the cells lining the space on here are cube-shaped cells. This is a simple cuboidal epithelium. Simple cuboidal epithelium is going to be responsible to allow secretion and absorption. It's going to be found in the kidney tubules, ducts, and the secretory portions of the small glands. Also on the outer surface of the ovary. If we're looking on here, I have a space on here, and the space is lined with cells. So it's epithelium. How many cell layers do you see on here on this side? I have one cell layer. So it's going to be a simple epithelium. What's the shape of the cells on here? What is the shape of the cells? They are tall cells. It's a simple columnar epithelium. Simple columnar epithelium. If you remember, we can find the simple columnar epithelium lining the gastrointestinal tract from the stomach all the way to the anus, to the anal canal. It's going to be responsible to allow absorption, allowing secretion of mucus and secretion of enzymes.